Hello friends, so today we will learn about the leukocytes. So leukocytes are nothing but the white blood cells which are present in our body is known as a leukocytes. So when we are exposed to variety of harmful pathogens such as bacteria, virus, fungi and parasites which can cause disease. So when we are exposed to very harmful pathogens which are virus, fungi, bacteria, so these may cause us to feel sick and they may affect our body. So to combat these pathogens we have a defense mechanism called as a leukocyte. So to combat these pathogens we have a specific defense system in our body. So this system is known as a leukocytes. Leukocytes are the white blood cells form a major part of this defense mechanism. So they may form a major part of this defense mechanism. So we will see about the classification. Based on staining properties, leukocytes are classified into granulocytes and the agranulocytes. So they are divided into two types, granulocytes and the agranulocytes. We will see about the granulocytes. So based on the color of granules on staining, granulocytes are further classified as neutrophils, eosinophils, basophils. So based on the color, by the differentiation of the color granules, these granulocytes are classified into three types: neutrophils, eosinophils, basophils, and the percentages of the neutrophils will be 60 to 65 percentage, and the eosinophils will be 1 to 4 percentage, and the basophil is 0 to 1 percentage in our body. Next, we will see about the agranulocytes. So, agranulocytes are of two types: lymphocytes and the monocytes. So, lymphocytes are 25 to 30 percentage, and the monocytes are 2 to 8 percentage. So, the percent mentions in the previous index indicates the number of each type of leukocytes in 100 cell is termed as a differential count. So these percent mentions in the granulocytes and agranules will be indicate each type of leukocytes. So these leukocytes in 100 cells will termed as a differential count. So this differential count will be useful for the diagnosis of various diseases, since a particular type of leukocytes is increased or decreased in disease condition. So these count will be helps in the in our, as we see, when we are affected by any pathogens or any viruses etc, so our white blood cells count will be increased, means eosinophils percentage will be increased. So like this it is useful to differentiate the count of the leukocytes is known as a differential count. Next, we will see the functions of the white blood cells. So, in the functions of the white blood cells, it has four major functions. First one is neutrophils and the monocytes are phagocytic. So, these are the phagocytic, and the eosinophils are anti-allergic and antiparasitic. So, eosinophils are the anti-allergic and the antiparasitic, and the basophils liberate heparin and histamine, and lymphocytes are involved in immunity. So these are the four major functions which we see in the WBCs. Next we will see about the leukopoiesis. So leukopoiesis is nothing but leukopoiesis developed from the pluripotent stem cells of the myeloid series. So these leukopoiesis will develop from the pluripotent stem cells of the myeloid series, and these cells gives rise to completed stem cell, are also called progenital cells. So these completed cells are also known as a progenital cell. So these are erased from the pluripotent stem cells, and the completed stem cells differentiate into various types of leukocytes. So these completed stem cell will differentiate into various types of leukocytes. Two linkages of leukocytes are formed. So two different linkages of leukocytes formed by the differentiates of the leukocytes. So the two differentiation linkages are myelocytic linkage and lymphocytic linkage. So these are the two major linkages. And next, the neutrophils, eosinophils, basophils and the monocytes are formed from the myelocytic linkage, and the lymphocytes are formed from the lymphocytic linkage. So these three, neutrophils, eosinophils, basophils and monocytes, these four are formed from the myelocytic linkage, and remaining one lymphocytes are formed from the lymphocytic linkage. Next we will see about the myeloblasts. So myeloblasts are nothing but these are the large cells 12 to 18 mu in the diameter. So these are the large cells which are 12 to 18 mu in the diameter, and they have round nucleus with 1 to 5 nuclei. So these have round nucleus with 1 to 5 nuclei and non-motile and do not contain granules. They are actively mitotic. So these myeloblast cell divisions occurs by mitotic divisions, and the cells are the large, and the diameter will be 12 to 18 mu, and it has a round nucleus, and it has 1 to 5 nuclei. So these are the non-motile and they do not contain granules. These are the myeloblasts. Next we will see the promyelocytes. So promyelocytes are nothing but the cells are slightly larger in size. So these are larger in cells will be slightly larger in size, and they have round nucleus. So they contain round nucleus. The nuclei decreased in the number. So these will be decreased in the number, and the chromatin begins to condense. So here the chromatin will be begins to condense. As we compare to myeloblast, the same thing we will see that it has round nucleus, but as we see here the nucleus will be 1 to 5 nucleus, but here the nucleus will be decreased. And extra point is added to this one is the chromatin begins to condense. Here we can't see any chromatin. So they have promyelocytes will are larger in size, and they have round nucleus, and the chromatin begins to condense, so the nucleus will be decreased in the number. Next we will see about the myelocytes. So myelocytes are nothing but the cells multiplied, the cytoplasm becomes less basophilic. So when the cells will be multiplied, the cytoplasm becomes less basophilic. So and the granules appears in the cytoplasm. So when the cells are multiplied, the cytoplasm will be basophilic, and the granules appears in the cytoplasm. The size of the nuclear decrease and the nuclear disappear. So when the size of the nuclear decrease, the nuclear will be disappear, and the chromatin condense further. So as we compare to the promyelocytes, here also the chromatin begins to condense, and the myelocytes will be also the chromatin further condense. But as we see, the nuclear will be round and decreased in the number, but here the nuclear will be disappear, and myelocytes will be occur by the mitosis is reduced but still condense in this stage. So mitosis will be occur, but it reduced to continue at this stage. And next we will see about the metamyelocytes. So metamyelocytes are the nucleus develops lobes. So here the nucleus will be develops the lobes. As we see, the nuclei disappear, but here in myelocytes the nuclear will be disappear. As we see here, the metamyelocytes, the nucleus develops lobes, and the cells stop multiplying and starts maturing. So here the cells starts maturing in the metamyelocytes, and they becomes fully mature and functional to form the leukocytes. So the metamyelocytes will become fully mature and functional for the leukocytes. Here we can see the full cell of the leukocytes, and the formation of granulocytes myeloblast takes around 10 days. So the formation of the granulocytes from the myeloblast takes around 10 days, and the monocytes are formed from the myelocytic linkage. So these monocytes will be formed from the myelocytic linkage. They are produced from the small completed stem cells called neutrophils. So these monocytes will formed from the myelocytic linkage, and they are produced from the completed stem cells of the neutrophils. So from the neutrophils the monocytes are appears. And next, the monocytes require 6 days for the formation. So the monocytes will be required for the formation of the full cell is 6 days. Next we will see about the lymphocytic linkage. So in lymphocytic linkage we will see about the lymphocytes. So lymphocytes are produced from the bone marrow and processed in the thymus. So these are produced from the bone marrow and they are processed in the thymus. So they have finally lobed in the peripheral lymphoid organs such as lymph nodes. So they have peripherally lymphoid organs. So lymphatic lineage means lymph system. So lymphoid organs are loaded in the peripheral lymphoid organs such as lymph node, spleen, nostrils, and the lymphoid tissue and the intestine. Next we will see about the lymphoblasts. So lymphoblasts are the large cells. They are 12 to 20 mu in the diameter with around round nucleus. So they are the large cells and 12 to 20 mu in the diameter. So they have round nucleus and the cells undergo division from large lymphocytes. So these cells will undergo division for the large lymphocytes and the large lymphocytes. So as we see in lymphocytic linkage, the lymphocytes will be about three types. So lymphoblast, large lymphocytes, small lymphocytes. When we do in practical counting of the WBC, we can see large lymphocytes and the small lymphocyte differentiately. So the large lymphocytes in this function they stop dividing and only mature. So they will stop dividing and they can only mature and they appear in the circulation. So they appear in the circulation. And next we will see about the small lymphocytes. So they are the formed from the fuse and maturation for the large lymphocytes. So small lymphocytes are formed for the fuse and maturation of the large lymphocytes. So next we will see about the regulation of the leukocytes. So regulation of the leukocytes means leukopoiesis is increased in the specific proteins colony stimulating factor, interleukins and prostaglandins. So these leukopoiesis will be increased in the specific proteins. So anything cell can need proteins or stimulating factors etc. So these leukopoiesis is increased in the specific proteins colony stimulating factor, interleukins and the prostaglandins. Next the protein formed by the SALIC gene. So these proteins are formed from the SCL gene, SALIC genes, and is responsible for the production of the pluripotent stem cells. So these stem cell leukemia is responsible for the production of the pluripotent stem cells. Next, the colony stimulating factor is the protein formed by the T lymphocytes, fibroblast, macrophages and the epithelial cells. It is of the three types. So as we see, the colony stimulating factor is necessary for the leukocytes. So it is also a protein produced from the T lymphocytes, fibroblast and the macrophages and lymphatic tissue. So and these are all of the three types. So SCFGM stimulate the production of the neutrophils, monocytes, eosinophils, erythrocytes and the megakaryocytes. So colony stimulating factor GM stimulates the production. So it helps in the production of neutrophils, monocytes, eosinophils, erythrocytes and megakaryocytes. Next we will see that the colony stimulating factor G, so it will stimulate the production of the neutrophil only. And next we will see colony stimulating factor M, it will be helps in the production of the monocytes. Next we will see about the internucleins. So internucleins means a hormone like substance regulating immune response. So it is a hormone like substance which helps in the regulating immune response. And the interleukin 1 to 3 and -6 convert pluripotent stem cells into counted stem cells. So interleukin -1 to -3 and -6 will be convert the pluripotent and uncounted stem cells into counted stem cells. And next the prostaglandins, cholesterol and adrenocortic hormone play a major role in control of leukopoiesis. So the things that will be play a major role in the intraproclines means the cholesterol and the adrenocortic hormone, and which they play a major role in the control of the leukopoiesis. So to here the leukopoiesis is completed. Next we will see about the morphology of leukocytes in another class. So thank you friends, please subscribe and comment if any mistakes or any doubts in the comment section. Thank you friends, please subscribe and like my channel.